With the recent COVID-19 pandemic, you've probably heard of PCR before, but have you ever wondered what it actually is? PCR stands for polymerase chain reaction, a tool used to make multiple copies of a gene through repeated heating and cooling. In short, PCR can clone specific DNA strands. Its ability to make billions of copies of a DNA strand gives it many applications, including research and genetic studies, forensic analysis, and medical diagnostics. The PCR process probably sounds quite familiar to you if you watched our DNA replication video. The first step of PCR is identifying the DNA sequence, or gene you want to replicate, and placing primers, short fragments of single-stranded DNA that match the start and end of the target gene, in the DNA pool. The second step is to heat the DNA pool to make the double helix DNA into single strands, separating the two strands. By lowering the temperature, the previously inserted primers can bind, or anneal, to their complementary sequences, marking where transcription should start and take place. Now, the enzyme Taq polymerase binds to the primer inserted earlier and begins transcription. The Taq polymerase adds complementary nucleotides to the strand, creating two pairs of the targeted DNA segment. Taq polymerase is not the only enzyme that can transcript DNA. For example, our body does not use Taq polymerase. Taq polymerase is used in PCR because of its high heat resistance, which is important considering that PCR requires high temperatures. This cycle is repeated through the heating and cooling of temperatures to create numerous copies of the desired DNA over a short period. Now that you know what PCR is and the process, I'll demonstrate what this might look like in the lab. As a recap, these are the materials needed in a PCR procedure: DNA gene to duplicate, nucleotides to make duplicate DNA, primers matching the ends of the target gene, Taq polymerase, and a reaction buffer for adjusting pH levels. Sometimes, scientists combine the common reagents in the reaction to form a master mix, which includes Taq polymerase, dNTPs, which are nucleotides, and a reaction buffer for pH, reducing the number of pipetting steps and risk of error. Aside from the materials needed in the process of PCR, you will need the following lab equipment: thermal cycler, centrifuge, PCR tubes, micropipettes, and Eppendorf tubes. Before starting, get a fresh batch of pipette tips to avoid cross-contamination. To avoid cross-contamination, always use a new tip for a new substance. Another thing to mention is to always open the Eppendorf tubes with both hands. It may be tempting to open it with one hand, but this may cause the material contained to spill and ruin the experiment. Now that I've covered the important stuff, let's get on to the actual process, starting with the mixing of material. In the background, you can see me pipetting each material from Eppendorf tubes into the PCR tube using a micropipette. A micropipette is a pipette that allows for 1/500th of a milliliter precision when pipetting. The PCR tube is a plastic tube that is specifically designed to be used in a thermal cycler. Make sure to pipette the Taq polymerase last, as this starts the reaction. Once you have every single component for PCR together, you will need to spin the mixture in a centrifuge to concentrate everything and eliminate any droplets on the wall of the tube. Depending on the radius of the centrifuge, the desired RPM will vary. It's now time to start the process of repeated heating and cooling. The thermal cycler, sometimes known as a PCR machine, is a device that allows you to do repeated heating and cooling. A program should already be pre-programmed into the thermal cycler made for PCR. This process should take a couple of hours. You now have hundreds of millions of the gene you wanted to duplicate. This now concludes the process of PCR.